Lisa is an internationally acclaimed statistician and biostatistician. She's a professor in Department of Statistical Science, Computer Science, and the Dalai Lama School of Public Health, Division of Biostatistics at University of Toronto. She is Tier 1 Canada Research Chair in Genome Data Science and Director of Canadian Statistical Science Institute, Ontario. Lisa's research interest covers a really great variety of topics, which focus on the development of novel statistical approaches to analyze and integrate multi-omics data to identify genetic contributors to complex human disease. And Lisa, so the stage is yours. Thank you. Welcome.
Thank you very much. Thank you for the invitation. I'm looking forward to chatting with everybody today. So if you have any questions, I can't see them, but people will interrupt me and I'll be able to address them if you want as I go through, or in the Q&A at the end.
Yeah, so you can type your question in the box, or you simply raise your hand. Okay, thank you.
Great, thank you. So so I'm going to talk about genome data science and focus on some tools and some approaches to understand GWAS loci. Really, my goal is to give sort of a background on our perspective of genome data science and all the aspects that are involved. Here you can see our beautiful city of Toronto, and here's the main university. And this is the downtown campus. We have 3 campuses, and we have many affiliated research institutes that also are carrying out, doing a lot of research in data science as well. And so it's one big exciting research community. I just wanted to let you know a little bit about Toronto and what we're doing, the environment that we're operating in. So Toronto is North America's 4th largest city, and it's considered one of the world's most diverse, the world's most diverse city, with greater than 50% of its population born outside of Canada. So it's really exciting environment to be doing genome research and research in general. And of course, then there's the University of Toronto, which is very large and has greater than 14,000 first-year students. And these are from 1,000 different municipalities globally. And so at the University of Toronto, we have the Data Sciences Institute. And the Data Sciences Institute here, this is the picture that I just showed you in the beginning of the downtown campus, but we have campuses in the other cities in the bigger Toronto area of Mississauga and Scarborough. And the Data Sciences Institute is really a central hub and incubator for data science research, training, and partnership across all 3 campuses and all of the affiliated research institutes. And our goal is to accelerate the impact of data sciences across disciplines to address pressing societal questions, drive positive social change. And so how are we doing this? Well, I wanted to note that what we're really trying to do is focus on bringing interdisciplinary teams together, having statisticians, data scientists, be PIs with individuals in specific domains coming together to work on pushing fields forward using tools and data science, developing novel tools that can advance new disciplines like genome data science, and having individuals, students, trainees, training at the intersection of these fields so that we can train the next generation of data scientists. So once these collaborative research teams come together, then they have access to many different supports through the Data Sciences Institute at the University of Toronto, including Catalyst grants, and we're removing barriers to access public data by covering costs of data access. We provide research software development support, and I'll talk a little bit about how that's been important developing software for us, as well as seed funding for methodologists. And so check it out. And I just wanted you to have an understanding of the environment in which we're working in Toronto and in genome data science. So how we look at data science is we see it as including, everything from the collection of data, manipulation, storage, visualization, the extraction of information. And this is certainly from increasingly larger and more heterogeneous and complex data. And of course, the data sciences is plural. And so there's genome data science where the focus is on this genomic data and doing so in reproducible, fair, and ethical ways. So in any type of data science, such as genome data science, there are unique considerations. And so, some of the unique considerations that are of particular relevance and challenges in genome data science. Well, first of all, the goal really is that there's information in the DNA sequences that needs to be decoded, and that's really, we're trying to get insight from the sequence of the DNA, and that could be in one individual, that could be across individuals with a common trait or a disease. And so there's storage challenges given data sets are significantly growing. And there's lots of challenges, in sharing the data due to things such as the genome being, so identifiable. And so, so there's corresponding ethical responsibilities, and so working that out. So these are some of the unique challenges. Then, of course, there's unique tools and algorithms that we need to create, and computational platforms that we need to compute in that are specific to the genomic data that we're working with. And then we require advances in technologies and informatics, and these advances have generated really large biological data sets besides genomics, but of course transcriptomics, proteomics, metabolomics. So part of genome data science is also integrating these different types of data sets to better understand biological systems and to feed into precision medicine goals. And so these are some of the unique considerations in genome data science. So today, what I want to do is I want to talk a little bit about our experience in genome data science with some of those challenges, and really use the Canadian Cystic Fibrosis gene modifier study as our example. And so I'm going to talk about cystic fibrosis and that Canadian study. And I'm going to talk about how we generate genomic data in the context of that study as an example, how we prepare the data, the genetic analysis. And then I'm going to talk a little bit about some of the tools that we've been developing in Toronto in my group, and that is a tool for co-localization visualization and co-localization testing. And when I say co-localization, what I mean is, data integration with other omic data, as I mentioned previously, which is a growing challenge or opportunity, however you look at it. And so we're going to review that. So first, cystic fibrosis and the Canadian CF gene modifier study. So, cystic fibrosis is a genetic disorder. Of course, it's caused by the gene, the cystic fibrosis transmembrane conductance regulator. In order to have CF, you need a variant, a causal variant, a mutation from your mother and your father. So it's a recessive genetic disease. And cystic fibrosis, so 1 in 26 individuals carries the causal gene, but you need a causal variant, but you need two copies of a causal variant to have the disease. And 1 in 3,600 live births in Canada have CF. Now, cystic fibrosis is quite a complex disorder in the sense that the cystic fibrosis transmembrane conductance regulator, the gene that causes CF, well, that protein is expressed in many organs, and so you see disease in many organs. And so, some individuals have various severity of their disease across the different CF affected organs. The disease in the airways is what results in the most morbidity and eventually mortality. It's progressive, and really what happens is there's a buildup of mucus in the lungs, and they see cycles of infection with bacteria, for example, and cycles of infection and inflammation that eventually results in progressive lung disease. Now, we're in a very exciting time in CF in the sense that there are these new medicines that target the causal gene itself. So this is really precision medicine, in that, you know, different individuals have different causal variants in their CFTR gene. And these new medicines target the different variants that cause the disease. Now, we don't have CFTR targeting medicines for every possible mutation that an individual could have, but they do cover a large proportion of the different CFTR mutations. And so that's really exciting, but there are several things to still consider in that there are CFTR mutations. So there are individuals who have CFTR mutations where they don't have drugs that are available to them. And some of these drugs aren't effective for some individuals. And there is a variable amount of response to these drugs. Now, what I have here in this image is a triangle that shows many of the affected organs in CF. And the triangle is meant to reflect the complexity in what we hypothesize to be the disease etiology in that particular organ. So you don't have CF and you don't have CF disease in a given organ unless you have two CFTR mutations, which is the big black circle on the right. But as you move down in that triangle, so for example, disease in the exocrine pancreas looks to be, we hypothesize to be contributed to through CFTR mutations, of course, as well as other genes that impact the disease severity. Whereas in the airways, you know, this disease in the CF airways looks to be like a complex trait, like lung function, for example, in the general population, where you actually have a lot of other genes, we call them modifier genes, and those are reflected here with these pink circles, and the environment. So modifier genes, CFTR, the environment, they come together to create variability in your lung disease. And so what I've listed in here are, over the past 15 years, some of the genes that we have identified that contribute to disease in the different organs in CF, and have done so in the Canadian CF gene modifier study, as well as with our international partners. And so how do we do that? Well, of course, what we need is a really engaged participant population, a large cohort, deeply phenotyped, access to biospecimens. And so here's a beautiful map of Canada. And what's noteworthy here is that individuals with CF, they receive their care in specialized CF clinics, because as I just noted, you know, CF is unique in that it affects many different organs, and you really need specialized care adapted to the needs of these individuals. And so there are specialized clinics that are scattered across the country of Canada, and depending on the population, of course, you have different numbers of individuals. And so we have recruited 3,300 individuals from 9 provinces across Canada, and that's over 75% of the Canadian population who has CF. And you can see there's so many different sites here, and so a huge team of individuals that are recruiting, in the sense that they are consenting individuals into these genomic studies, and obtaining biospecimens and sending those biospecimens to us at the University of Toronto and the Hospital for Sick Children. So we also, as I mentioned, we don't just need these DNA samples or these biospecimens, but we also need very rich clinical information on individuals all across the country, thousands of individuals. So not just whether they have CF or not, but really understanding the depth and the severity of disease in their different organs. And so we're so fortunate to have access to a large disease-specific registry, the Canadian CF Registry. So it began in '73. It was in concert with the US CF Registry. And the data comes from all those participating CF centers. So there's 42 CF clinics across Canada, and the different centers, they submit data from each clinic visit, from each participant that comes into the clinic, into this registry. And so you have the cross-sectional, you have longitudinal data, so decades of lung function information about the bacteria that are colonizing their lungs, what CFTR drugs or other types of treatments they're on, the comorbidities that they have across their organs. Do they have diabetes? Do they have, were they born with an intestinal obstruction? And we even have, for example, whether they tested positive for a SARS-CoV-2 infection. And this registry is open to anybody who wants to apply to it for the purpose of research or clinical care through CF Canada. And so we have linked our biospecimens and our recruited participants to the CF registry. So we are able to link all of that information and have all of this information on those who consent. So just so you have a snapshot of, for example, what the Canadian CF population and what's in this registry look like in a given year. So this is the 2020 snapshot. So at this point, there was 4,300 Canadians with cystic fibrosis. So as I mentioned, we have over 3,300 that are recruited, consented into the CF modifier study. So that's over 75% of the population. And you can see that there are more than 18,000 clinic visits that are captured in the registry. You can see how many lung transplants there were, that 62% of the Canadian population are actually adults. And you can see things like their median lung function. So in children, for example, their median lung function is 93% that of a healthy child of a similar age, whereas in adults, their lung function is about 65% of a healthy individual. So I explained here where we get our data from. So this is from across Canada. We merge it with the CF registry to get the clinical information and to get an understanding of the CFTR drugs that all of these individual participants are or are not on. We get biospecimens, and these are sent to our team at SickKids and the University of Toronto. And then we extract DNA or RNA or whatever it is that we're interested in, and we get the omic data. So I'll talk a little bit about how this is generated: genome sequence, RNA sequence, genotyping arrays. But what is our goal? Well, our goal is to identify those genetic contributors in my triangle earlier so that we can build predictive models of disease severity, so how severe their lung function may be, and of their therapeutic response. Can we predict how they'll respond to different CFTR drugs? And they need new drugs? And then also to identify alternative therapeutic targets, so not just targeting CFTR, but also are there other targets that genetic information can tell us that we should be targeting through our genetic epidemiology studies and our genetic analysis. Okay, so how do we generate the genomic data? What does this look like? So I mentioned earlier there are many types of omic data. We can have the genome, we can have the transcriptome, we can have the methylome. So here's just a simple example of some of the data that we're collecting. So we collect SNP array genotype data, DNA sequence, RNA sequence. So what do I mean? So here's the phosphate backbone of the DNA, and here are particular locations across the DNA, so markers across the DNA, and these are single nucleotide polymorphisms. We call these SNPs, and they can have they're either an A, a T, a C, or a G at this location, and so we can genotype these using SNP arrays. Well, so these are just probe-based technologies that are probing the genome in certain locations all across all of the different chromosomes. But to create sort of a map of a certain number of genotypes, a certain number of SNPs across the genome. And we have the genotype of them. So one from the mom, one from the dad. So they're either say AT or AC or AG at these particular SNPs. Or, we could do DNA sequencing where we sequence all the different possible alleles here, not just at these single nucleotide polymorphisms, but we sequence the whole genome. and that's DNA sequencing. So we would chop up the DNA, we sequence all the letters in the fragment, and then we put the puzzle back together, and that would be the genome sequencing, the DNA sequencing. Alternatively, we may be interested in gene expression. So not what the sequence is in the genome, but how much, for example, protein there is of a particular gene. Now, when we're doing RNA sequencing, we're not measuring protein. We're using a messenger RNA as a proxy of expression, of protein expression. And we do that because there have been studies that show that they correlate well, although this is still a question. And so here we would take the mature RNA here. That's so this is the RNA that doesn't have any of the introns or intergenic regions. It just has the cDNA basically. So this is just the messenger RNA. And so we would capture that and we would just sequence it in the same way as we sequence the DNA. And then we can quantify each gene's mRNA level by basically just counting up the number of transcripts. And like I said, we use that as a proxy for gene expression. And so these are, I'm bringing these three to the forefront because these are what I'm going to talk about today. Now, you could use that gene expression data for many purposes. Differential gene expression analysis you may have heard of, we're not going to talk about it today. Or you can integrate the SNP genotypes or the genotype data with the expression data and construct and compute something called eQTLs. So you could look at whether a SNP, remember in the phosphate backbone, those SNPs, they could be a T, a C, a G, or an A. You could see whether this T, for example, is associated with more transcript, more mRNA, more expression than the A allele. So basically it's a phenotype. You're looking at whether this allele is associated with greater expression being the outcome, for example. And this is called an eQTL, an expression quantitative trait locus. So here in this case, a T is associated with high gene expression and an A is associated with low gene expression. So you're integrating both that genome array-based technology or sequence, the SNP, the genotypes with the expression. Okay, so in the Canadian CFG.
A question, sorry, from the audience. Sorry to interrupt you. It says, like, do you determine the SNPs from your RNA sequence data as well? If so, how good is this working?
No, so we don't. What we do is here we have our population genotyped on these arrays. So on the genotype arrays that I described up here. So we have almost the whole population has a genome-wide genotype array, and then subset of the population have RNA sequence. And so the intersection of that we can compute eQTLs for. You can also use public data for eQTLs, but so it's an excellent question, and here is sort of an illustration of a list of the different types of technology that we have on the Canadian CF gene modifier study.
Thank you.
So, for example, we have almost everybody genotyped using a genotype SNP array. We also have many individuals who have undergone who we've done whole genome sequencing of their DNA, including some of the latest new technologies in long read sequencing. I'm not going to talk about the genome sequencing today. I'm going to focus on the array-based technology and the RNA sequencing, where we have brushed the noses of individuals with CF to get their nasal cells, because that's a model of the airways in CF. So the expression in the nasal cells has been shown to reflect the expression in the upper airways. So I'm going to just talk about those two today. Okay, so data preparation. So another part of genome data science, of course, is just preparing the data, manipulating the data. You know, and of course, we talked about the clinical data preparation, and that's that actually is a big part of getting reliable, heritable phenotypes. And in fact, we did spend a long time trying to work out the best, most heritable phenotype for lung function, for example, because you have lung function, but you also have you have longitudinal lung function, and you have individuals who are where there's significant mortality with age, so it can get complex. But so there's that component. And then there's the quality control of the genotype calls, the genotype data. There's quality control that we have to do for a given sample. I mean, we have to understand the ancestry of the population, the genetic ancestry of the population that we're studying, because that can lead to, for example, we know that individuals in different subpopulations have different types of CFTR mutations, so different causal mutations of their disease. And so that certainly could introduce confounding if we don't know and adjust for genetic ancestry. We need to estimate the relationships in the data, because we can't assume everybody's independent and have a full set of a bunch of families in the data. We need to, that will dictate the statistical model we use, and that we need to adjust for non-independence. And then, I mentioned that we have data, genotype data, and they're from all kinds of different platforms. What that means is that we have, yes, we do have array-based technology on the whole sample, but we used different SNP chips, which means there's different sets of SNPs all across the genome. Was there a question?
Yeah. So there's a question says, did you collect family and pedigree information on all the cases and in your prediction model?
So I'll show you in a second, but we do have siblings. What we did is we only enrolled individuals who have CF into the study, and those are the ones who are receiving their care in CF clinics. And so when it is often the case that you have multiple siblings in a family who have CF, and so we have those individuals in the data set. But we don't have their parents. But we would only have their parents if their parents also had CF. There are some situations where we do there. And then in terms of genotype imputation, what is that? Well, we have many individuals, they're genotyped on different arrays, which means there's a different set of markers along the genome that individuals are genotyped on. But if we're going to analyze the data, we need to make sure that we have a common set of variants across the genome. So we need to impute the genotypes so that we have that common set across all sets of individuals. So we'll talk a little bit about that. So I'm not going to go over all of the quality control, but I do want to note here, I put this clock here because it's often the take home and the exciting part is, of course, the results of the genome-wide association study or some of the statistical analysis that comes after that. But almost 99% of the time is spent on the data preparation, manipulation. And so it's very important, and also a very active area of research, some of these different components of the quality control. So what do I mean when I say quality control on the SNP level? Well, here's, let's say, all of the different genotyping platforms that we use, which means different sets of SNPs that were genotyped across different groups of individuals. And so they have different numbers of, not only different sets of SNPs, but of course also a different number of SNPs. And so we have to look, okay, well, so, is the call rate high on, for all of these SNPs? Because if not, we should remove those, because that might indicate some sort of systematic error in the calling of those SNPs or the probes that were used on the array. So these are some of the sorts of things. Do we have duplicated variants? We should remove those. What about on the sample level, meaning the participant level? Well, sometimes an individual's genotype vector has a lot of missing data in it, and that might suggest that there's some problems with the DNA, for example. Or there's a mismatch between the sex that we estimate from their genetic data and what's in the CF registry. And so that could suggest that there's a sample mix-up. So, this is some of the quality control that we need to do on the genotype level as well as on the sample level. And what about inferring genetic ethnicity? Well, lots of ways to do this, also an active area of research. But basically, what you would do to try to understand the genetic ancestry makeup of your population, you could merge your samples with public data of known ethnicities, such as what's made available in 1,000 genomes or HapMap3 populations. And then you could calculate principal components for the merged samples. And you can, so here's PC1 against PC2, PC1 against PC3, and 2 and 3. And here's the color coding of the different populations from known data. And the CF samples are coded in red. And so what you can see is the CF samples, for example, are clustering very closely with the European samples from the HapMap, as well as the Gujarati Indians from Texas. So you see some samples as well from the CF samples that are clustering with that. So you start to get a picture of what the genetic makeup, ancestry makeup is of the sample, so that we can adjust for that. And there's an active area of research as well, and the best way to analyze that data, to how you should adjust. Should you adjust using the principal components in a regression model? Should you be using mixed models? Are there other ways to do it? So also an active area of research. But most important is to understand and know and be aware of the genetic ethnicity of your population. You also need to understand whether there's relatedness, and thank you for preempting that question earlier. So if you're going to do a regression model, for example, linear regression model, you're assuming that every observation is independent. But if you have siblings, of course, that's not the right model to be using. And so you need to estimate the relationships in your data from the genetic information. And so this is sort of classic output from a package called Plink, where there are some specific rules that are recommended, but this is also an active area of research, the best way to estimate relationships and how you can get better and better at estimating more distant relationships. But there are rules here, so you can see that, for example, the black dots correspond to independent individuals. Each dot is a pairwise relationship. And so purple represents full siblings in our data set. Here, the red are parent offspring relationships, and the blue are MZ twins. And so, of course, what we need to do is we need to just take one MZ twin, one of the pair of MZ twins for our analysis. We need to make sure we have a random effect for our siblings, families. We can treat the independents as independent, of course. So this helps us to identify the model that we're going to use when we're doing our genome-wide association study. And last, genotype imputation, also a major area of research, best ways to impute. So here, what we have is you have SNPs here from that genotype array, for example, the genotype haplotype, so from each allele from the parent. And then you take a reference population that's been whole genome sequenced. And so you have their haplotypes, you have all the possible haplotypes so that you know what different types of alleles you could fill in here. And the whole goal of this is of course to have more variants to analyze, but also to be able to analyze a common set of variants across all of those samples that we have when, I mentioned earlier that they're genotyped on all kinds of different arrays. And so you fill those in probabilistically, so that you fill the genotype array data in from this reference haplotypes to get these imputed haplotypes. So now you can analyze these particular variants, not just the ones that were genotyped here in blue. So I mentioned an active area of research. Some work that we were interested in doing ourselves is trying to understand, well, how do you pick this reference population where you impute? And so, we were doing some work trying to create hybrid references. So is it useful to use some sequence from the population that you're interested in studying? For example, the CF population, because we know that they probably come from unique subgroups. It's potentially some of our founder populations. And so, we constructed a hybrid reference where we put together the sequence from CF individuals as well as public data and tried to compare the imputation quality when we construct these hybrid reference panels. And we do observe that we get improved genotype imputation quality when we include in-sample in-sample sequence. Okay, so we know the relationships in our family now, in our data. We know the genetic ethnicity of the individuals that are in our study, and we know, and we have done the genotype imputation, so we have a common set of SNPs to analyze across everybody. And we cleaned our data to the best of our ability. And so now we can go ahead and start to think about the GWAS, the genome-wide association study, the genetic association study. So we merged efforts with our international partners. So I told you all about the Canadian CF gene modifier study, which you now know is a population-based study trying to capture the full population of CF individuals in Canada, and same with the French study. There's also a study out of the University of North Carolina that we collaborate with, as well as, excuse me, a twin and sibling study from Johns Hopkins University. So in the French study and the North Carolina study, there's no relationships in the data. These are all individuals who come from independent families. Whereas in the Canadian CF gene modifier study, I showed you, we have individuals who are independent, but we also have siblings in the data set. And in the Johns Hopkins study, it's a study of twins and siblings. So we have to remove a twin, keep one, and we have to account for the sibling relationships. And so that dictates the type of model that we might use for lung functions, quantitative trait, linear regression in the independent samples. We use linear mixed models in the family, in the studies where there's family relationships. Here you can see that there's different arrays in each one of these different studies, which complicates things. So what do? We did basically 13 GWASs, so 13 platform, so a genotype array by study combinations. We're analyzing over 16 million SNPs after we imputed to that a common set of 16 million SNPs. We code the SNP as additive, so you have 0, 1, or 2 reference alleles. We adjusted for principal components for population structure for our genetic ancestry in each one of these 13 platform by study combinations. That's how we chose here to adjust for population structure. And then we meta-analyzed all 13 of these groups together using fixed and random effect meta-analysis. And here's the GWAS. Here are the results. So basically, on the x-axis, you have the chromosomal location. On the y-axis, you have the minus log base 10 p-value from the association here of the SNP effect and an additive coding. Each dot represents one SNP along the genome and its association with lung function on the minus log base 10 p-value scale. And here is the genome-wide significance line. So any locus genomic region that has variants that are above this genome-wide significance level are assumed to contribute to CF lung disease. They reach the genome-wide significance criteria. So we have several loci. Now notice that, I'm not putting one gene here. I have a list of a couple genes. And that's because genome-wide association studies, they identify loci. They don't identify genes. They don't identify mechanisms. They just identify SNPs that are markers of regions. So how do we figure out? Yes.
So a question from the audience: what software did you use for the meta GWAS analysis?
Yeah, we just coded it in R. It was in 2015, but there are several packages now that are common. I don't remember the most common, but yeah, we didn't use a particular software package at that time.
Okay, thank you.
So the question is, how do these loci contribute to lung disease? What's the responsible gene, right? We have no idea. So here, let's take this locus here on chromosome 5. Okay, and we're just going to zoom in. So each dot here on the right corresponds to one of the dots in this region over here. And what you can see here is, first of all, that there are many genes in this region. We have no idea what the responsible gene could be or what the responsible mechanism. Why do we see an association in this region? What else do you see? Well, so here's this purple SNP is the lead SNP. It's coded, it's the one with the smallest p-value that we observed. And then the color coding represents the linkage disequilibrium or the correlation between the lead SNP and each one of these other SNPs in the region. And so really it assesses whether what the difference is in the probability of carrying the two alleles together minus the product of their marginal frequency. So just that independence, the correlation. So what you can see is that for variants that are in linkage disequilibrium that are correlated, of course, you see a very similar significance level. But what you can see here is there's two regions of association that seem to be independent. Here you have the blue, which show very little linkage disequilibrium with this lead SNP, very little correlation. Okay, so this looks like a very complicated locus. This is a large locus. There's a big area of linkage disequilibrium here, and these two peaks represent something called allelic heterogeneity, potentially that there are different causes or more than one contributing variant or locus or to lung disease, and that's why we see this region. Okay, so why do we see an association? Why do we see a GWAS significant variance here? Well, the first thing you look at is that the associated SNPs, they're actually non-coding. They are not in the coding regions of genes. And often what that means is that potentially the causal variant or variance in this locus could affect gene expression, how much expression you have of a given gene, but we don't know which one. And in fact, if you take a look in public databases of the top SNPs in this region, what you would see is that they are associated with gene expression of SLC9A3 in individuals who don't have CF. So that's of interest. This is a very big region, as I mentioned, 280 kilobases. But there are 5 genes in this region, and all of the genes, like 5 major genes in this region, but all of the genes, all but AHRR, are expressed in lung epithelia. It's also important to note that we know something about SLC9A3, this particular gene here. It has been studied in other phenotypes in CF, mostly the intestinal phenotypes, and it has been shown to associate with intestinal disease in CF. So it's a good candidate, but we really don't know anything. And the role of SLC9A3 in the lungs is not well known at all. Okay, so the questions that arise obviously are, is the relationship between these associated SNPs and CF lung disease somehow mediated through gene expression? Are these variants impacting gene expression, and the impact on gene expression is what then impacts lung disease? And if so, what is the responsible gene? So how are we going to figure that out? Well, the first thing we need to do is start to visualize all of the data that's accessible to us. So not just the GWAS SNPs, so these dots represent the GWAS locus here that we've seen on all the other slides. But now what we've also done is we've taken, so this gray line here represents the EQTLs for SLC9A3 in this region, where we sort of fit a line through it, and this is in the lung. So what do I mean here? So we remember that an EQTL takes a SNP and looks at its association with expression of, and here in this case, SLC9A3. And we can compute a p-value that's on this y-axis here. Okay, and that p-value here, we sort of draw a line through windows so that we can visualize what the p-value p-values of the EQTLs for 9A3 look across this region of these SNPs. And so this sort of, and what you want to see here is whether these EQTL p-values co-localize. Do you see a similar pattern between the association between the SNP and the phenotype, and between the SNP and the amount of SLC9A3 gene expression? Because that tells you that the same SNPs that are contributing to lung disease are also contributing to 9A3 gene expression, and that that could be the smoking gun, that could be the mechanism of action. And so we created this visualization tool where you can take a look in this region and you can hover over the SNPs and you can see what the SNP name is and you can see what its log base 10 p-value is. And then you can also take the SNPs that would contribute to the EQTLs and you can find out what the EQTL p-value was. You can see if that same variant that is a GWAS SNP, well, what is its p-value for an EQTL? And so you can start to dissect the region and understand whether the same variants are contributing. So here, what you can see is that this particular SNP, it has an EQTL that has, you know, a minus log base 10 p-value somewhere in the order of 8 here. So it, you know, it does have evidence of an EQTL, but it's certainly not the strongest evidence of an EQTL in the region. The strongest evidence of the EQTL are actually in regions that aren't at variants that aren't even present in our GWAS data set. And so what that tells us is that potentially this is, as we thought, a very complex region. So this is how we can visualize the region and get a feel for whether we do indeed have co-localization. So we do see these yellow dots that are the EQTLs from which this gray line was drawn. And so we have this visualization tool and we want to see, okay, we do see some overlap in the EQTLs and the gene expression. It's not complete overlap, but we need to test it. So how do we test for co-localization? So there are a lot of methods out there to test for co-localization. There's Bayesian approaches, and they assume that you want to find whether the expression, the EQTL, and the GWAS have shared causal variance. And here are some of the methodologies that are out there to use them. All excellent. There's Mendelian randomization approaches, which try to determine whether the gene expression causes the lung disease. So they use a Mendelian randomization framework. And then there's other approaches that try to take the gene expression, take the genetic, the genotypes and impute the gene expression, and then look at association between gene expression and the outcome. And so we were looking to just use some of these methods, which would be great. However, the problem is that none of these methods could apply to our situation. And that's because, as I mentioned, as you saw, this is a region of very high linkage to disequilibrium, and many of those methods don't perform well in regions of very high linkage to disequilibrium, especially when you're trying to make assumptions of causal variance, for example. We're in a region of allelic heterogeneity. So remember the two signals. We constructed the GWAS p-values from a meta-analysis with and without related individuals. And so it was not clear whether the other methods out there could address the summary statistics that are constructed from meta-analysis. We have related samples. And because we were working with the consortium, we didn't have individual level data. Yes?
Sorry, a question from the audience. What is the name of the tool for co-localization?
Yeah, so I'm going to talk in a little bit about a tool that we developed for co-localization, but some of the tools that already exist for co-localization testing, so testing if there's that overlap, here's some of their names: eCaviar, Coloc, Coloc2. My references are here. The Mendelian randomization type co-localization methods, SMR. And here's some of the gene expression imputation approaches: PredictScan, TWAS. So these are all co-localization. So there's a lot of co-localization methods out there. But I'm going to talk about one that we had to develop, and that's because I already went through our situation, right, with the high linkage disequilibrium, the allelic heterogeneity, the meta-analysis, and all of these complicated factors. The methods out there, there wasn't a method that could address all of them. So we had to develop our own. And the other complexity here with co-localization testing is actually that the null hypothesis is composite. And many of the other methods also address this composite hypothesis, but it's very important to note because when you're trying to think about whether you have controlled the type 1 error rate, you have to think about the composite null. And what do I mean there? Well, that you could be under the null hypothesis in several ways, right? You could have no GWAS causal variance, and you could have no EQTLs in the region. So then certainly there's no co-localization. You could have no GWAS causal variance in the region, but you could have EQTLs, but certainly not, that's under the null hypothesis of no co-localization. And the opposite, you could have GWAS, but no EQTLs, and you could have both in the region, but they don't overlap. So there's different causal variants, for example, that contribute to each one. And these are all under the null hypothesis. And so when you're constructing a test for co-localization, you have to make sure that you are controlling the type 1 error under this composite null hypothesis, all 4 cases. So here's how we took a very simple approach to trying to construct a test statistic to do co-localization that would enable us to address all those issues I talked about. And this work is done by Fan Wang, who just was a PhD student in my lab and just defended her thesis last week. And so that's very exciting. And so this work is published in over a course of her studies. And most recently in the American Journal of Human Genetics. So let the Z's be the GWAS summary statistics, let's say the log-based 10 p-values or the Z statistics. And here we have the EQTL summary statistics. Now let's say we dichotomize the EQTL evidence. So here you have this blue line here, and they represent the minus log-based 10 p-value. Here, sort of best fit, this is just a cartoon of the EQTL evidence. And you can see that it's co-localizing with the GWAS evidence, which is the red, and it's higher. And so let's say that you dichotomized it. We're not going to do this, but let's say you did dichotomize it. And you say that any EQTL that has a minus log-based 10 p-value greater than 4, let's say that that's an EQTL and it's 0 otherwise. So then what you're really interested here, heuristically, is that you want to know whether the average Z statistics, the average GWAS summary statistics, are higher in regions where you have EQTLs versus regions where you don't have EQTLs. So that's the thinking of co-localization. And then, of course, you'd be interested in, say, a one-sided test, because only this positive difference suggests co-localization. So that was our thinking about a co-localization test statistic. And we can extend that to the use of a continuous measure of gene expression, which is what we do here. And so this is our test statistic here, where A is a diagonal matrix, where the AJs are just this EQTL p-value, for example, summary statistics. And so we're able to show, when we use this particular test statistic, that this sigma here is the genotype correlation matrix. That's that LD matrix that I was talking about earlier in the plot, the correlation between the variants. And then, so this test statistic is just a mixture of chi-squares, where these DJs are eigenvalues from this particular matrix here. And so we can control the type 1 error rate under all of the null hypotheses, except we do have some inflation here under H03. So that is when there's no EQTL, but there is some GWAS. And so to fix that, like other methods have done, one option is to just require that you have a high EQTL threshold. But we wanted to avoid that. And so we constructed a 2-stage test where we formally test whether you have EQTLs in the region with this test statistic. And then if it passes that stage, then you just perform that simple sum test statistic. And this analytically controls the type 1 error rate under the composite hypothesis, so that's great. And you can also control for a multiple gene by tissue pair, so you can compute the co-localization test statistic for all the genes in the region. Remember, there were many, and you can adjust for multiple hypothesis testing and still control the type 1 error rate, the family-wise error rate. And so through simulation, we can show that we have family-wise error rate control under multiple hypothesis testing. And to deal with the meta-analysis approach issue, we could incorporate summary statistics, and we just adjust for the covariance between the summary statistics. And also we can incorporate overlapping samples by using methodology that already exists in the literature by using a decorrelation approach. Yes?
Another question from the audience.
Mhm.
Without access to individual level data, from what population do you estimate the genotype correlation matrix? And what impact does it have?
Yeah, such a good question. So we did do some sensitivity analysis using an LD matrix that's from 1,000 genomes, for example, and it doesn't seem to have a huge impact. And I think that many other studies have also shown that you can use an external LD matrix. However, of course, it is best if you do have access to the LD matrix from the sample that you're analyzing or interested in. I think in the paper, we do address that question and we discuss it to a fair degree. In the software, which I'll show you in a second, we have an option to just pick an LD matrix from 1,000 genomes from different subpopulations as well. Or you can upload your own LD matrix in that software. And so in terms of power, so here's a situation here where you have obvious co-localization. It's very simple. You have one GWAS peak, you have one EQTL peak. And in this scenario, some of the other software does better. They're designed for this purpose. Colloq2, for example, does very well in this particular situation. And here is the SS2 approach here with the red triangles. When you get to a situation where you start to have some allelic heterogeneity, then that's where SS2 really seems to protect you and perform well in those scenarios. So all of these are situations where you have allelic heterogeneity, whether it's the EQTLs or whether it's the GWAS, and that's where the SS2 really does well. And so, and of course, certainly you can tell in a given situation potentially what the scenario, what scenario you are operating in. But once you get to a scenario where you have a lot of genes, a lot of tissues, and you're calculating across all kinds of things, all kinds of different gene by tissue pairs, then you could definitely be in a scenario where you're significantly worried about power and the potential of allelic heterogeneity. So that we implemented the visualization tool as well as the co-localization testing in some software called LocusFocus. You can find that at locusfocus.research.sickkids.ca. So this is what you input is the GWAS p-values and then the p-values from any secondary SNP level data set. You can also choose EQTLs from GTACs across tissues, and you can just automatically pick that from a drop-down menu, and you can input an LD matrix that you define, you obtain from somewhere else, or you can pick an LD matrix from a set of choices from 1,000 genomes. And the output are co-localization and heat map plots for and the visualization. We also implement colloq, so you can also use that method as well, assess that method and compare it. And so here, this is the sample output that you see the overlap, and you can, like I mentioned before, you can hover over variants to explore the region and the comparability across the SNPs. And here is, and I'll just, and the heat map, and I'll just zoom in a little closer now so that we can just talk about the application to 9A3. So taking the SLC 9A3 locus across all of the genes at the locus, we construct the stage 1 test. Remember, that just tests whether there's a EQTL, whether there's EQTL evidence for this particular gene in the lung. And you can see that we do see some EQTL evidence for AHRR, for 9A3, and for XOC3. And so those particular genes EQTLs for those genes would move on to stage 2, where we do the co-localization testing with the GWAS summary statistics. And so here, I just want you to focus on the two that seem to show some evidence of co-localization, which was the SLC 9A3 gene, as well as the XOC3 gene. And so that helps us to hone in on what is the responsible gene, and also lets us know that there is indeed some co-localization in that the mechanism by which the SNPs are contributing to lung disease looks like it could be through gene expression. And that, that helps guide us in further follow-up functional studies. You could actually take a look across, you don't just have to look in the lung, of course, that's what makes sense here. That's what we were studying. This was lung disease, but just to be comprehensive, you can look at all possible cell types, and you can look at all possible reported genes in the locus over a different region, and you can get a heat map. And this is the co-localization p-value, color-coded, and you can see that there's evidence of co-localization across lots of tissues for many different, these three particular genes. So you have a good view of the whole region. I just wanted to say, that's the co-localization methodology that helps us try to dissect and understand that GWAS locus. But, this framework is quite simple and can be extended to all kinds of different questions. I won't really go into it too much, but, I mentioned that XOC3 and SLC9A3 seem to show some expression, some co-localization. So you might ask, well, is there more evidence of co-localization from one gene than the other? And so you could contrast the co-localization evidence, and that could be straightforward, where you remember this sort of heuristic model where we categorize the gene expression and look at that. You can look at that difference as well here. So you have the difference in the gene expression between the two genes, and you integrate that with the GWAS test statistic. And under the null hypothesis of no difference in co-localization, you can compute that, you can construct that test. And so, so that's an example. Another example is you could use that same test statistic, and you could ask, okay, let's not look at EQTLs, let's look at other GWASs, GWAS summary statistics. And is there cross-trait genetic correlation or genetic contribution? So does lung function and FEV and in the CF and the general population correlate? Is there overlap in the genetic contributions to lung function in CF and in non-CF? And so you could certainly use a statistic to do that and summarize it across more than one locus. So a locus on chromosome 7, a locus on chromosome 3, or genome-wide. So just to sum things up, and then I guess we can move to the Q&A if there are any more questions. But in terms of co-localization, so we proposed a summary statistics-based co-localization visualization and testing tool, and you can find that in a series of papers that we published over the last 3 years. And what it does is it integrates GWAS summary statistics with any secondary data set, but potentially EQTL summary statistics across multiple gene by tissue pairs. And it doesn't require assumptions that there's a single causal variant, and that's really why under allelic heterogeneity it works out quite nicely. It's quite robust when there's allelic heterogeneity. It leverages the linkage disequilibrium, and so it's powerful in regions of this allelic heterogeneity. And we're able to incorporate some summary statistics from meta-analysis and from statistics calculated using related individuals. So it could address the challenges that we saw and the complexities in the summary statistics from GWAS that we constructed. And like I said, it's broadly applicable to integrate any SNP-based summary level data, contrast co-localization evidence across genes and tissues. And so in conclusion around genome data science, so as technologies advance, we have new challenges in genome data science, of course. There's the storage and the compute and quality control, data integration and harmonization, and new challenges, new questions are constantly arising as we see new technologies such as long read sequencing and we need comprehensive, well-defined, engaged participant cohorts and interdisciplinary teams to work on these projects, which is why I wanted to talk early on about our approach to interdisciplinarity at the Data Science Institute at the University of Toronto. And to note that the loci we find in GWAS, they're located in complex regions of the genome. We look at SNPs and we use those as a tool, but there's lots of different types of variation that can be contributing to disease that's difficult to sequence or genotype, or often not captured. And so these require novel methodology development, so lots of room and area to work in. And the availability of large and even public omic data introduces opportunities for data integration techniques and new insights. And lastly, GWAS, of course, is just the beginning of a long journey. Well, in genome data science in general, the collection of these cohorts, the multi-omic data generation, is just the beginning of a long journey to understand the genetic contributions to traits. And lots of work for us in genome data science and statisticians to do that work. So I just want to note the huge group that are involved in the Canadian CF gene modifier study in Toronto, but across the country in Canada. This is a very big effort that's been funded by the CF Foundation and the Canadian Institutes of Health for a really long time. And we have a CF center at SickKids as well, with amazing basic scientists that contribute a lot when it comes to collection of the biospecimens and follow-up studies functionally. The patients and the families are so engaged and are constantly being asked to participate in studies and spend so much time and effort on research studies, and just we're so thankful to have a engaged cohort, and the great trainees and staff. In my lab, Fan developed the co-localization, Naeem the software and the visualization. I want to comment on Katie and Julie, who are doing the patient recruitment and scraping of noses and collecting of the blood samples, and the Genome Center, of course. All of our funders. And with that, I'll take any more questions if you have them.
Thank you very much.